Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss a topic called Allopolyploidy. First, I am going to give the definition of Allopolyploidy. Allo means other. Allo means other or different. Then polyploidy means an organism which possesses more than two basic set of chromosomes, and that organisms are called as polyploidy. So allopolyploidy means an organism which possesses more than two set of basic chromosomes derived from two different species, and that condition is called as allopolyploidy. Then allopolyploidy can be developed by interspecific cross. So due to that interspecific cross, that F1 hybrid may be sterile in nature. F1 hybrid may be sterile in nature. So we have to restore the fertility of that allopolyploids. So we have to treat that allopolyploid with the chemical called colchicine. So by treating the plant with the colchicine, we can restore the fertility of that allopolyploid. Then allopolyploid can be developed between closely related species. Allopolyploids can be developed between closely related species. Then in our book, they are given two examples for allopolyploids. They are Raphanobrassica, that means raphage. Radish plus cabbage, they are called as raphage. Then the second example is triticale. So this is the first man-made ciliate called triticale. So now we are going to discuss the two examples, Raphanobrassica and triticale. First, I am going to explain the Raphanobrassica, how that scientist made a cross and how he produced that raphage. So please wait a minute. Let me tell about the Raphanobrassica. Raphanobrassica was developed by Russian genetist G.D. Karpchenko in the year 1927. He made a cross between Raphanus sativus and Brassica oleracea. So Raphanus sativus is the scientific name for radish. Cross between Brassica oleracea. Brassica oleracea is the scientific name for cabbage. So he made a cross between Raphanus sativus and Brassica oleracea, and two of them are diploid in nature. That means 2n is equal to, and it contains 18 chromosome. Likewise, in the Brassica oleracea also diploid in nature, and it contains 2n is equal to 18 chromosome. So cross was made between the two individuals. During gametogenesis, meiosis takes place, that is the reduction division takes place. So that somatic chromosome get half and they are produced, or they are referred as gametes. So they get half, so they are reduced into n is equal to 9. The same condition occurs here also. So n is equal to 9. Then this gametes contains the chromosome. Here also they contain chromosome, but for our convenience I am going to write the chromosomes. So the gamete contains 9 chromosomes, and that chromosomes are denoted by capital R, capital R, capital R, capital R, capital R, capital R. So here it contains 9 chromosomes, which denotes capital R. That chromosome name or the chromosome names are capital R. Then here it contains 9 chromosomes and they are denoted by C. The chromosomes are denoted by C. So C, capital C, capital C, capital C, capital C, capital C. So here it contains 9 chromosomes and they are denoted by capital C. So fusion of gametes takes place. Fusion of gametes takes place. That means n plus n is equal to 2n. Then 9 plus 9, it produces 18. So 2n is equal to 18. 2n is equal to 18. And they are, this young one is considered as F1 hybrid. That young one is considered as F1 hybrid. I already told you, in Allopolyploidy, the F1 hybrid may be sterile. So this young one is a sterile young one. The young one is sterile young one because it contains 2 different set of chromosomes. It contains 2 different set of chromosomes. So the Raphanus sativus contains R chromosomes. The Brassica oleracea contains C chromosome and they are totally different. So that is why they are sterile in nature, because the reason is during meiosis they fail to pair. They fail to pair because the 2 chromosomes are different in nature. So during meiosis they fail to pair. That is why they are sterile in nature. That is why they are sterile in nature. So how can we restore the fertility of that young one? So we have to apply that chemical called colchicine. That chemical called colchicine. So by applying the chemical colchicine, we can restore the fertility of that plant. So we apply that chemical to that plant. So after that application of colchicine, after the treatment of the colchicine, the plant chromosome get double. That plant chromosome get double. That means 2n is equal to 4x. How many x? 4x, because it contains 2 set of chromosomes. So it get double. 4x is equal to 36 chromosome. 36 chromosome, and they are considered as fertile ampiploids. Ampidiploids, and they are considered as fertile ampidiploids. What is called ampidiploids? What is called ampidiploids? Because this organism possess 2 set of chromosomes from the Raphanus sativus and 2 set of chromosomes from Brassica oleracea. So that organism that is considered, this is a raphage. It is a raphage. So raphage contains 2 set of Raphanus sativus chromosome and 2 set of Brassica oleracea. So it contains both the individual chromosome. This organism contains both the individual chromosome. That is why they are called as ampidiploids. Then here what takes place? Here capital R, capital 9 chromosome is present. After the treatment of that colchicine, it get double. That means capital R, 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 7, capital R, capital R. Here capital C, capital C, capital C, capital C. So after the treatment of colchicine, the chromosome number get double, and in F1 generation, consider this is the F1 generation. So in F1 generation, the pairing of chromosome takes place. During meiosis, the pairing of chromosome takes place. That is why they are fertile in nature. And here they are tetraploid in nature. Here they are tetraploid in nature because it contains 4x. Because they contains 4x. So raphage is a tetraploid. Allopolyploid. Okay. Tetraploid in nature. Then that scientist failed to produce raphage because he expected this plant to exhibit the root of radish and the leaf of cabbage. That means the raphage has to exhibit the root of radish and leaf of cabbage. But he was totally disappointed because that condition is vice versa. That condition was in vice versa condition. That means that raphage produce root of cabbage and leaf of radish. That plant produce root of cabbage and leaf of radish. So he failed to produce the raphage in successful manner and totally he was disappointed. Then I am going to explain the second example, triticale. Triticale is the first man-made cereal. Triticale is the first man-made cereal. Depending upon the ploidy level, triticale can be classified into 3 types. They are tetraploidy, hexaploidy and octoploidy. Tetraploidy means crosses made between diploid wheat and diploid rye. That is called tetraploidy triticale. Then hexaploidy means the crosses made between tetraploid wheat, triticum durum. Triticum durum means Marconi wheat and rye, diploid rye. Then octoploidy means they contain 8 set of chromosomes. They contain 8 set of chromosomes. So crosses made between hexaploid wheat, triticum aestivum. Triticum aestivum is called as bread wheat, commonly called as bread wheat. So this is the wheat we are using to make chapati and bread. So triticum aestivum is called as bread wheat and made a cross between rye. So in this video we are going to discuss hexaploidy triticale. So the cross made between tetraploid wheat, triticum durum and diploid rye. So let me figure out that cross which was made between the triticum durum and rye. So please wait a minute. Let me explain about the triticale, how the triticale is formed. Triticale is a hybrid plant which possesses both the characteristics of triticum durum and sisal cereal. Triticum durum means the Marconi wheat, sisal cereal means the rye. The triticale contains high amount of protein like the Marconi wheat and it possesses high amount of amino acids, lysine, contains high amount of amino acid lysine like rye. So both these characters are present in the triticale. Both these characters are present in triticale. So triticale bears or possess high amount of protein and high amount of amino acid lysine. Now I am going to explain the cross, how the triticale is formed. So the cross is made between, we are going to make a cross between triticum durum and sisal cereal. So triticum durum is the scientific name for Marconi wheat. Sisal cereal is the scientific name for rye. Then triticum durum is in tetraploid nature, and sisal cereal in diploid nature. So 2n is equal to 4x is equal to 28 chromosomes. So they are in tetraploid nature, tetraploid condition. Rye in diploid condition. 2n is equal to 2x is equal to 14 chromosome. Then gametogenesis, they undergone the process gametogenesis. So a reduction process takes place, that is meiosis takes place, and they get reduced into, the gametes contain n is equal to 2x. They contain 2 set of chromosomes and they contains 14 chromosomes. How many chromosomes? 14 chromosomes. Here n is equal to x and they contain 7 chromosomes. Fusion of gametes takes place, results in the development of F1 hybrid, and it shows 2n is equal to 3x and 21. 2n is equal to 3x plus 21, and they are sterile in nature, and it shows triploidy condition, because it contains 3 set of chromosomes. So they are in triploidy in nature. So the F1 hybrid is sterile. So we have to restore the fertility of that sterile plant by applying colchicine, the chemical colchicine. So after the application of colchicine, the chromosome get doubled. The chromosome number get doubled. So 2n is equal to 6x is equal to 42. Now it restores its fertility. So now the plant restores its fertility. So 2n is equal to 6 is equal to 42, and it results in the production of triticale. So triticale is the first man-made cereal, and they are in hexaploidy condition. So that's all about the triticale. So still I am going to explain one topic, that is significance of ploidy. So please listen to all the topics. Let me tell you the significance of ploidy. So here I have written 5 points about the significance of ploidy. So the first point tells us many polyploids are more vigorous and more adaptable than diploids. So vigorous means they are strong and they are very active and they are very energetic. And they can resistant to any kind of disease. So they can withstand to any kind of disease or any kind of environment. So many polyploids are more vigorous and more adaptable than diploids. Then many ornamental plants are autotetraploids and have larger flower and longer flowering duration than diploids. So ornamental plants are mainly grown for maintaining aesthetic sense, and they are autotetraploids, and they have larger flower. So that flower can attract many insects, and that flower can attract the people who have seen that flower and having their longer flowering duration than diploids. Then the third point, autopolyploids usually have increase in fresh weight due to more water content. For example, seedless watermelon, potato, tomato. So they are increased in size and they contain more fresh weight due to more amount of water content. For example, we can take it that Bengaluru tomato. So it contains more amount of fresh weight because it contains more amount of water content. Then aneuploids are useful to determine the phenotypic effects of loss or gain of different chromosome. So aneuploids are useful to determine the phenotypic effect of an organism by loss or gaining of different chromosomes. Then the last point is many angiosperms are allopolyploids, and they play a role in an evolution of plants. So the allopolyploids creates new types of plants because that cross was made between two different species. So the allopolyploid produces new kind of organism, new kind of plants, and it leads to evolution of plants. So that's all about today's video. If you have any doubt, please let me know. Thank you very much for watching this video patiently. Thank you very much.